What are some modifications made by the microbe to their peptidoglycan in order to protect themselves from lytic cleavage? Well, in microorganisms, they have several kinds of modifications made on peptidoglycan. And really important thing to remind yourself is that microorganisms are unicellular organisms. So, which means that whatever they do really always talks about, am I going to die or am I going to be alive? So they would always try to find a way to survive. And it's pretty clever because microorganisms, they don't even have brain, but instantly they know how to survive. So here we go. Actually, here I outline all the modifications that occur in peptidoglycan. Well, really not all, but the major points that I need to know for my exam. And I thought it'd be helpful if you know what kind of modifications occur in peptidoglycan in general. And the actual answer for this question is the last 2 bullet points here. I will describe in detail, but I just wanted to mention these 3 here. So usually in gram-positive, trimming process occur, trimming of pentapeptides into tetrapeptides by D-decarboxypeptidase. And pentapeptide is the original form, well, after they've incorporated newly synthesized peptidoglycan into the cell wall, it's actually in this form, pentapeptide. And then even further trimming occurs from tetrapeptide to tripeptide using L-decarboxypeptidase. And you can, well, the cell also decreased the average glycan chain length by using a lytic N-glycosylation to make the size about 35 to 40 disaccharides. Once that happened, then they add major outer membrane lipoprotein called Brown's lipoprotein covalently to the sacculus by LDTPases. This can also act as signal later when peptidoglycan gets old or mature or it needs to be replaced. And for me, I think of this step as regulation process. It doesn't really help much for a microorganism towards protecting themselves from lytic cleavage. And now here is the actual answer for the question. Really important. O-acetylation at C6 hydroxyl group here of the N-acetylmuramic acid residue protects bacteria from lytic trans-glycosylase cleavage, which means that it's preventing lysozyme to break down peptidoglycan of the microorganism. Another way to survive is S-lactam formation in gram-positive, and this is usually seen in endospore-forming gram-positive microorganisms for their peptidoglycan formation. And this helps the cell to protect themselves from host immune system by having these types of modifications. N-deacetylation, well, it allows microorganisms to evade the immune system or the lysozyme. Here, really important microorganism. I don't know if you've encountered, but Listeria is a very clever microorganism because they are phagocytized by macrophage, but they are able to prevent, or do they form phagolysosome complex, but Listeria somehow evades lysozyme binding to the cell membrane inside the macrophage, and then this kind of sneaks into the cytoplasm of the macrophage. So, well, why is that important? Because macrophage is only effective if microorganism is outside of macrophage. But since Listeria is now inside the cytoplasm of macrophage, there is no other immune system or immune cell that's like, oh, hey, there's there's listeria inside your tummy macrophage, so you better watch out. No, there's nothing like that. Once they get into the cytoplasm, they're gonna be happy and they're gonna live like, oh yeah, be happy and be merry. So this is a very smart way for these types of microorganisms to survive. And that's why you don't want these types of microorganisms to get inside your macrophage in the first place. So stay healthy. And here's the next one, N-glycosylation. This is a process adding oligosaccharide or glycan to nitrogen on asparagine. And adding polymers to C6-hydroxyl group also helps these spore-forming peptidoglycan forming gram-positive microorganisms from lytic cleavage. Anything when you have a lot of bonding or thicker or more cross-linking, the more complex it is, it gets difficult for immune system to act on you. So that helps microorganism to survive. And here is a really important point I wanted to note. Pentapeptide is common in gram-positive, but it is not found in gram-negative so far. So which means that whatever I mentioned up here, the trimming process and a sort of regulation-like process does not really occur. Well, this here, trimming pentapeptide here, does not occur in gram-negatives. So far, we haven't seen it yet. Alright, see you next time.